So now if we talk about non-membrane bound organelles, so this means that these come in direct contact with the cytosol and they do not have a membrane surrounding the structures. So the ones that we'll talk about: ribosomes, the cytoskeleton, centrosomes and centrioles, cilia and flagella, and microvilli. So with ribosomes, big thing that they are doing is protein synthesis. We have two main types of ribosomes. We have first that are going to be floating and are going to be unattached within the cytosol. So here is an example of ribosomes. So they're very small structures and they're going to again be in the cytosol, but they're not attached to anything. The second type that we have are going to be fixed ribosomes, and these are going to be on the outer surface of the rough endoplasmic reticulum. All right, so if we look at this microscope image here, what you can see is the endoplasmic reticulum, and it has two sides to it because it's membrane bound, and these little dots or speckled appearance is going to be the ribosomes that are attached. So that's why we have the rough endoplasmic reticulum, because the ribosomes are attached here. All right, next we have the cytoskeleton. So this is where we find proteins that are going to be within the cytosol, and they're going to either be solid filaments or hollow tubes in one special case. So big thing here is that the cytoskeleton is made up of three components: the microfilaments, intermediate filaments, and microtubules. So micro, you might imagine, these are going to be very small. Intermediate, these are going to be in the middle, and then the tubules are going to have the hollow tubes. All right, so let's start with the microfilaments. And so here is an example of the microfilaments. And these are made of actin. And so these are going to be intertwined and have this helical appearance to them. This is important for maintenance and for change of the cell, because if you remember, cell is not going to be very static, it's going to be very dynamic. And this helps with muscle contraction and also for cell division for all of our cells. And what we have next are the intermediate filaments, so slightly larger than the microfilaments. Big thing here is that they're for structural support, and they're going to help to stabilize the position of the cell in between other cells. Very important when we talk about epithelial tissue later on in the semester. All right, lastly, we have the microtubule, and so these are going to form a hollow tube that will have proteins that are going to arrange themselves in that fashion. These are going to actually radiate from the centrosome, and they have many functions. They help to fix organelles in place because they are slightly larger than the microfilament and intermediate filament. They also help to maintain the shape of the cell and also give it a rigid structure so that it doesn't all dissipate at once. We'll also have movement of organelles for cells because our cells are living things. Proteins are going to move all about, and we'll have an increase in cell motility. So for some of our cells, we have cilia and flagella, which we'll talk about in just a couple slides later, and this is for movement of cells. Example for with flagella, this would be like sperm. And last but not least, but help for chromosomes to move during cell division when we talk about mitosis. The next non-membrane bound organelle, centrosomes and centrioles. I'm just going to start backwards and talk about centrioles first. So these are going to be sets of 9. So 1, 2, 3, 4, 5, 6, 7, 8, 9 microtubules that are in triplets. So 9 by 3 microtubules. Okay, these are important because they're involved in the organization of microtubules, and then also are attachments for the chromosomes during cell division. And so this is what will cause then the chromosomes to migrate when we go through mitosis. Now, centrosomes are going to be a pair of centrioles at right angles. So we're going to have a perpendicular arrangement to them. Okay, under 3D it looks a little bit different. Okay, so now we're looking at the centriole but times 2. All right, and so they are again going to be perpendicular to one another. Also, big thing here, if we look at this cell, here's the nucleus, here is going to be your centrosome. These are going to be adjacent to one another. Very important because once we go into mitosis, this is going to help to pull the cells together and the chromatids. All right, next structure, let's talk about cilia and flagella. So these are going to be projections of the cell that are going to contain the cytoplasm and microtubules because these are going to be capable for movement. So as you can imagine, not all cells will have cilia and flagella. So let's talk about cilia first, okay? So here is our example of cilia, and here's a cool microscope image of cilia right here. And these are going to be grouped on cells that are going to move objects across their surface, all right? So for instance, we have this in our respiratory tract, and so this will help move mucus out of our lungs so that we can expel it out our nose, okay, so stuff doesn't get trapped. And so they're going to be like little finger-like projections that help to move things along. We'll also find cilia in females, and this is found within the oviduct, so that it helps to move the egg along, going from the ovaries into the uterus. Then what we have, another example, is the flagella. There is only one example in the human body, and it's only found in males because this is found in sperm. This is going to be a very long extension, and this is typically singular, all right? So mostly made up of microtubules, and so it has this whip-like fashion and movement so that it can help to propel a cell. And so you can imagine it can move in long distances because of its very long extension for the microtubules. All right, one last non-membrane bound organelle. We'll talk about microvilli. So micro, you can imagine these are going to be something that's very small, and these are going to be thin microscopic projections that are going to extend from the plasma membrane. So similar to that of cilia and flagella, however, it's going to be much smaller. And so you can imagine too, not all cells are going to have microvilli. So the big difference between microvilli and cilia and flagella is that microvilli do not move. So they do not have anything traveling down it. Rather, their purpose is to increase surface area. So if we look at this image here, I'm highlighting these individual microvilli. You can imagine this is increasing the amount of surface area. And so important, like if we have this within the small intestine so that we can absorb food stuff and increase then the nutrients that are going to the cells.